Plant hormones and their commercial uses. Plant growth can only happen in specialized undifferentiated cells, meristem, which is found in the tips of roots and shoots, apical, lateral buds, cylinders of roots and shoots, lateral, leaves which branch off lateral buds, intercalary. Plant hormones, aka plant growth regulators, help coordinate growth, development, and responses to stimuli by affecting the division, elongation, and differentiation of cells. Despite not being produced by an endocrine gland, plant hormones are still considered hormones as they are transported away from the site of manufacture to the target cell, although some stay in cells that make them exert their effects in the cell. Plant hormone action. They are produced by specific cells. They have a specific shape. They bind to receptors on target cells which are complementary in shape to the hormone. They are carried by diffusion, active transport, or mass flow in the phloem or xylem. Some hormones have different effects on different tissues. Plant hormones can work in synergy with other hormones, amplify each other's effects, or antagonistically cancel each other out. Hormones can be commercially important and can be used by humans to produce ornaments or food. Auxins. Auxins are a group of plant hormones that cause the elongation of target cells. They do this by stimulating enzymes to weaken the cell wall, in order to add more cellulose to the cell wall and more phospholipids to the plasma membrane, enabling the target cell to grow longer. Auxins act synergistically with cytokinins and gibberellins. Recent studies have shown both synergy and antagonism with ethine. Auxins act antagonistically with cytokinins and abscisic acid. The most common auxin is indole 3-acetic acid, IAA. Auxins stimulate the active transport of H+ ions into the cell wall. As they are pumped in, the auxin-enhanced H+ pumping lowers the pH of the cell wall, activating pH-sensitive enzymes, E.G expansion, which separates crosslinks from microfibrils. With the cellulose loosened, the cell can elongate. In low doses, auxins can be used to stimulate cuttings to grow new shoots. This is often sold commercially as rooting powder, which is often used by florists. They can also be used to make unpollinated flowers develop fruits, often seedless fruits, and stop trees from dropping their fruit before harvest, preventing bruising, which can lead to wastage as the fruit cannot be sold in an aesthetically poor state. In high doses, they can be used as a weed killer by causing rapid growth. Ethine/ethylene. Ethine is a gaseous hormone which inhibits cell elongation, promotes seed germination, and helps to ripen fruit. They also cause apoptosis to make leaves fall off, abscission, for example in deciduous trees. 1. The concentration of auxin decreases as the leaf ages. 2. Cells in the abscission layer, where the leaf is attached to the branch, start producing ethine. 3. This stops auxin production in the abscission layer. 4. This weakens the abscission layer so the leaf falls off. Commercially, ethine is typically used to stimulate fruit to ripen. Delicate or soft fruits may be damaged in transport quite easily, so fruit can be harvested unripe, and therefore harder, transported and ripened artificially by ethine. This also allows for perfectly ripened fruit and allows fruit to ripen at the same rate. This helps increase shelf life and reduce wastage. Cytokinins. Cytokinins are involved in the morphogenesis of bud formation. They can cause the growth of lateral buds, leaf expansion, delay leaf senescence, aging, and promote chloroplast developments in leaves. They also have a role in the stomatal openings of some plant species. Cytokinins promote cell division. Commercially, this is important as they can be involved in the mass production of crops. By promoting chloroplast development, they can prevent yellowing. They can also prevent ripe fruit from aging, reducing wastage and increasing shelf life. They also help control tissue development and micropropagation. Plants enter senescence due to shorter days in autumn, drought, or lack of mineral ions. These stimuli cause lower production of auxins, which causes lower production of cytokinins as they work in synergy. This slows the rate of mitosis, leading to much less growth and repair. Some tobacco plants have been genetically engineered to make cytokinins and so live longer. Abscisic acid. Abscisic acid inhibits germination and growth, bud growth, and causes the stomata to close when little water is available, reducing gas exchange and water loss by transpiration. Gibberellins are a group of hormones that help plants grow by stimulating cell division and elongation. They can also stimulate germination and break dormancy. Commercially, they can be used to make unpollinated flowers develop fruit and are often used to produce seedless fruits. Farmers may apply gibberellins to shorter plants to stimulate growth or to seeds to induce germination. They can also delay ripening and aging in fruit to create larger produce and improve their shape.